this one slide is basically going to summarize without much detail all of section 3. So we have antigen-presenting cells, which is going to display antigen fragments from that pathogen. Helper T cells are triggered from that and release cytokines, or cytokines, however you want to say it. And the fact that the cytokines are released are going to trigger two different responses from our body. It can trigger a humoral immune response, which is where antibodies will be made to help with those pathogens. And humoral immune responses is a response that protects our blood, our lymphatic fluid, by neutralizing and eliminating toxins. The second type of response will be cell-mediated immune response, which I'll get more into that on this slide in just a second. So humoral immune response in the fluid of our body is where B cells are activated by those helper T cells. B cells differentiate and become more specific and divide into plasma cells, and plasma cells are able to secrete antibodies, which are tags for destruction for that pathogen. So this is one simple, well not simple, but one diagram kind of illustrating one type of response thanks to that antigen presenting cell and helper T cells. The other that I just mentioned a moment ago, cell-mediated immune response, which includes a different kind of T cells that are going to destroy host cells and kill by making toxic proteins, which I'll get more into that in a little bit. But anyways, helper T cells, again, they release cytokines. It's going to activate, those are going to activate cytotoxic T cells, which are the cells that identify. So how does an antigen-presenting cell have the antigen on the outside of it? Because this antigen-presenting cell is not the pathogen itself. So a different type of cell, like a macrophage or macrophage, it engulfs the pathogen and becomes the host cell. So it's like infected with this bacteria or this virus intentionally to be able to display that antigen fragment. So that way the T cells, the helper T cells, are able to trigger those two different responses, the humoral and the cell-mediated. So just to quickly say it, antigen-presenting cells engulf the pathogen, break up the molecules, and displays the foreign molecules on the surface, which you can see by the diagram. When it's being presented on the outside by this MHC molecule, and it's just the name of the molecule that you don't need to know anything else about it other than it holds that antigen fragment from the pathogen. It presents it to the helper T cell. And again, it binds the antigen receptor of the T cell, binds to that antigen, and then that activates that helper T cell. Here's another kind of silly illustration showing you what I just said on the last slide using a dendritic cell. To remind us, a dendritic cell is still a cell that originates in our bone marrow. Our stem cells produce them. And I think on, I don't know what section it was, I said that they can be in our epidermis, which is true because our epidermis comes into contact with so many pathogens. But dendritic cells can also be located in our digestive system, obviously our skin, as I already mentioned, in our airways, anywhere there's going to be contact with these pathogens, because dendritic cells are able to engulf. So they're also can be known as phagocytes or phagocytes. And they eat the bacteria or pathogens that they come into contact with. And once they eat or engulf that bacteria or pathogen, parts of the bacteria, the antigen part, goes to the surface of that dendritic cell, and the phagocyte presents the antigen to a helper T cell, which you can see in step number 3. And when it's presented on the outside, that helper T cell is activated, which you can see at the bottom of the screen. The role of the helper T cells is to say, help, 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 help to the body. And it does that by releasing the cytokines, which, to remind you, are a protein that is a signaling molecule. The signaling molecule is going to signal or activate the B cells and those cytotoxic T cells. Now, when it activates cells, it is going to activate in a way for clonal selection. So it's going to select for that cell to produce thousands of clones of itself, those cells that are specific to that antigen. And from there, those can be effector cells that are being cloned. They can be memory cells. Effector cells being B cells, which are going to make plasma cells, which again make the antibodies, or memory cells, which are also B cells, which keep a memory of that pathogen in order to quickly assemble the antibodies in a faster secondary immune response. A little bit more detail about clonal selection, making clones and B cells that are going to have different jobs. So the first thing is the antigen binds to the B cell receptor, which is activated by those cytokines or cytokines. Millions of clones of the B cells are made specifically for that specific antigen that was presented to it. Now, in the millions of clones of B cells, plasma cells will be derived from a undifferentiated B cell. These plasma cells produce antibodies that are specific to that antigen. And another type of B cell that can be derived from those millions are memory cells. And memory cells are going to have the same receptor, antigen receptor, and they're long-lived, and they're going to quickly mount a very quick immune response for future infections with that same pathogen. Another diagram showing you clonal selection of a specific B cell. So at the top of the diagram, you can see there are different B cells that have differences in their antigen specificity. So the first one, again, has like a green antigen receptor. The one in the middle has a purple, and then the one on the right has a blue. All different kinds of antigen receptors. And please again note that these are not realistic diagrams. It is not just one antigen receptor per cell. There's hundreds of thousands. But these cells are showing you that there's that only one is going to be triggered, basically. So when the antigen fits and is presented to the correct cell and antigen receptor, then there is that colony, the cloning taking place. So it is going to be replicated, and of all the different kinds that are replicated, some of them will become specialized to be plasma cells, which will secrete those antibodies that will match up with the antigens on the outside of the pathogens. Some of the other B cells will remain as memory cells, keeping that receptor in order to identify contact with that same antigen. Remember, B cells give rise to plasma cells, which are the cells that produce the antibodies. Looking at the diagram, step 1, the pathogen is engulfed by an antigen-presenting cell, something like a dendrite or a macrophage. The pathogen is broken down, and the outside of the antigen-presenting cell will carry that antigen fragment and signal a helper T cell. The antigen receptor of the helper T cell will bind with that antigen fragment of the antigen presenting cell. That will release cytokines. The cytokines are signaling molecules which will trigger the B cells to be cloned. Two types of B cells will be cloned, memory B cells and plasma cells, again giving rise to and making antibodies, which will basically tag the pathogens to be destructed. So this is what antibodies do. They mark pathogens for destruction. Antibodies bind to antigens on the different kinds of pathogens. They prevent them from being able to enter into other host cells. So when your body gets infected, the virus needs to be able to somewhat attach. It can either insert itself into your body cells or inject its genetic material into your body cells, and this will prevent it. Those antibodies clogging up the outside will prevent it from being able to do so. It also will tag them for destruction. So they have that antibody on the outside, and the body recognizes that antibody being attached to a bacteria or a virus or just pathogens in general, and they will be destructed by a T cell, a cytotoxic T cell. The majority of what I've discussed this far has only considered first exposures to a pathogen and a first exposure to an antigen on the outside of that pathogen. So this is a summary showing you after the first exposure, the pathogen and antigen on that pathogen is taken in by antigen-presenting cells, which is going to stimulate the production of helper T cells, cytotoxic T cells, B cells, all in that first exposure. And there's a little bit of a difference when there's a second exposure and the fact that we already have immunity to the certain types of bacteria that we may come into contact with. First exposure and making these B cells, helper T cells, and the cytotoxic T cells can take 2 to 3 weeks, let's say, the whole process in eradicating the bacteria or the infection. When we have a second exposure to the same antigen, we already have some lines of defense made in our immune system. We already have memory B cells. We have memory cytotoxic T cells as a part of the cell-mediated immune response, and then those memory B cells are in our fluid, part of the humoral antibody-mediated immune response. And because we already have those cells, then automatically plasma cells can be made much quicker. They can be cloned a lot faster and secrete those antibodies at a much more rapid rate. And then the same thing goes for the cytotoxic T cells. They automatically can release their chemicals to break down those pathogens that much faster. Now, how much faster? Anywhere from 2 to 5 days faster as opposed to 3 weeks with the compared to the primary first exposure. This is a graph showing you immunological memory comparing a first exposure over a period of time compared to the antibody concentration. So a couple things to note about antibody concentration. The antibody concentration is going to increase when there needs to be the antibody. We've been exposed to a pathogen, bacteria, viruses. So it's going to, our body, or plasma cells, need to make those antibodies. once there isn't that exposure anymore, the line is going to decrease, indicating that antibodies don't need to be made any longer. But then there's a secondary exposure to that pathogen. So immediately there's a sharp rise in antibody concentration, and it's going to be quick. It's not going to take as long. And then instead of the line completely decreasing back down, our body maintains that antibody concentration, a stable level. So that way, when and should we come into contact again with whatever pathogen it is, our body has antibodies already made for it, and again, it'll be broken down that much quicker. So just quickly kind of summarizing, primary immune response, first exposure to the antigen. Thankfully, we make memory cells. In that secondary immune response, repeat exposure faster and a much greater response. There's two different types of immunity and how you can get immunity. Okay, two different types, active versus passive. Active immunity is when you're exposed to an antigen, or the exposure to an antigen leaves behind those memory cells. So this would be when you were actually being infected. And immunization, or a vaccine, is active immunity, but it's induced, it's artificial. It's where you're inducing that immune memory to a non-pathogenic microbe or toxin. Passive immunity is from a mother breastfeeding and passing her antibodies to the baby. Even though our immune system is awesome, it does so many different things and it's so complex and wonderful, there are disruptions and problems sometimes with our immune system. One disruption to our immune system are allergies. Allergies is our hypersensitive response to harmless antigens. Antigens on the outside of certain kinds of cells, like pollen grains having an antigen on the outside of it, and we are hypersensitive to that. Another disruption to the immune system are autoimmune diseases. So where our bodies lose tolerance for our cells and they turn against molecules in the body. Cytotoxic T cells attack and damage our own cells. So examples of different kinds of conditions that people can have that are autoimmune diseases: lupus, rheumatoid arthritis, type 1 diabetes, multiple sclerosis. Another disruption to the immune system is HIV. This is a virus that infects helper T cells, and from infecting the helper T cells, it compromises our immune system, and AIDS is where it's a syndrome of having a severely weakened immune system because of the HIV virus. Also, some types of cancers are caused by viruses. So viruses that have infected a person. Viruses are involved in about 20% of cancers, and there needs to be a vaccine developed against these viruses so that way people don't have to get cancer later in life. An example of that, and this isn't a for sure, much like all this stuff, hepatitis B. Hepatitis B virus, often there's an increase in probability that somebody may have liver cancer. So having the hepatitis vaccine and having it is something that can be preventative in terms of cancer later in life.